In this video, I will be discussing how the genetic code was deciphered. The first experiment was conducted in 1961 by Nuremberg and Mattei. The objective was to determine what amino acids were created by codons composed of only one type of base. They did this by using a homepolymer, poly-U, mRNA, which was added to a test tube containing cell-free extract. This extract contained the necessary components for translation and all 20 amino acids. The test tubes were then incubated, which caused translation to occur. After incubation, the protein was filtered and checked for radioactivity. The experiment was conducted 20 times, and each time a different amino acid was radioactively labeled. The only time a protein was formed was when the radioactive amino acid was phenylalanine. The experiment was repeated for poly-C, poly-A, and poly-G. At the conclusion of these tests, Nuremberg and Mattei found that poly-U encodes for phenylalanine, poly-C encoded for proline, poly-A encoded for lysine, and poly-G did not code for any amino acid. 3 of the 64 codons were discovered in this experiment. The first words of the genetic code had been deciphered. Then, in 1964, Nuremberg and a different scientist named Lieder sought to crack the rest of the genetic code by finding the remaining 61 codons. They conducted an experiment involving ribosome-bound tRNAs. This experiment sought to answer the question, what other matches between codon and amino acids could be determined through the use of tRNAs? In this experiment, they used very short mRNAs that contained all of the different codon combinations. The mRNAs were added to a mixture of ribosomes and tRNAs attached to different amino acids. The tRNA was then allowed to bind to the ribosome. If the tRNA bound to the ribosome, then it would be caught by the filter paper, while the unbound tRNAs would pass through. The filter paper was assayed to determine which amino acid was present. The experiment was repeated using a different radioactive amino acid each time. Using this method, Nuremberg and Lieder identified 61 remaining codons. As a result of this experiment, a coding dictionary was created. These findings paved the way for deciphering the sequence of the entire human genome.